Welcome back. In this segment, we're going to be looking at how insulin is genetically engineered. Remember that this doesn't only apply to insulin. It could be a different hormone that is being produced for human consumption. It could be vaccinations that are being produced. The process is identical. You might see this process in your exam paper, and what you'd be seeing is something like this. Maybe you have to provide annotations, maybe you have to describe the process, and it looks, when you see the whole process put in front of you like this, it looks hugely complicated. But what we're going to do is we're going to break it down into 9 steps that are pretty easy for you to follow. And if you have your piece of paper there, why don't we get started? The first step in the process is to say and to recognize that we have a human cell that has got a nucleus and a chromosome that has that insulin gene. And it's the correct insulin gene. It's a normal, healthy insulin gene. Remembering, of course, that the diabetic hasn't got the facility to produce that insulin. So we need to make insulin from a normal, healthy cell. We first of all are going to take that chromosome and we're going to cut out the insulin gene. We call that to ligate the gene. Ligate means to cut it out of its original chromosome. We're not interested in the hundreds of other genes that are on that chromosome. We're only interested in the insulin gene. And so we can use enzymes to ligate just that particular gene out of the chromosome. We take that gene and we're going to insert it into a bacterial cell. So we've got the human insulin gene. We now need to insert that human gene into a bacterial cell. So let's have a look at the bacterial cell that we have. And I mentioned to you earlier, focus on that plasmid, that little circular ring of DNA that exists inside the bacterial cell. That is the DNA of the bacteria that we are focusing on for genetic engineering. And what we do is we isolate the plasmid. We can take the plasmid out of the bacterial cell, and using enzymes, we can cut it open. And taking it out the cell would be step 3, and cutting it open would be step 4. We're then going to take that human gene and we're going to splice it in. We're going to take it and join it into that plasmid. So now the plasmid, once we tie up the 2 loose pieces of the plasmid, has got the human insulin gene inserted into it. We then take this modified plasmid and we reinsert it back into the bacterium. And so what we have now is a bacterium that's got its plasmid back again, but included in the plasmid is 1 gene from human DNA coding for the protein insulin. What we then do is we culture the bacterium. Now, what happens is the bacterium will undergo its normal processes. It will undergo its normal metabolism, where it produces all the things that it would normally produce. And it undergoes asexual reproduction. And as you know, and you would have learnt in grade 11, asexual reproduction of these bacteria in favorable conditions is very, very rapid. And so this whole process of genetic engineering is rather clever, because we've created a little insulin producing factory in this bacterium that reproduces itself. And so our factory just grows and grows. And while it is undergoing its own metabolism, expressing its own genes and making its own proteins, it also makes human insulin. The bacterium has no way of segregating or separating its genes from this new included human gene. It simply expresses the human insulin gene along with its normal genes that it would have expressed anyway. So let's say on that plasmid there is a gene for making the protein that covers the bacterial cell, that slime capsule. The bacterium will make the slime, but it will also make human insulin. It does not distinguish between the human gene and its own genes. And all the scientists have to do is collect and purify this insulin that will be in the bacterial culture. They take the human insulin out, they purify it, and they can then use that insulin in medication for diabetics. The diabetics can simply inject that insulin into their own bodies, and the insulin acts as it would in a normal non-diabetic. It is human insulin, it's not pig insulin, it's not calf insulin. It's the precise form of the insulin that the human body is adapted for. And this is this overall process of genetic engineering. Now, in your studies of genetic engineering, you possibly will encounter questions along the lines of, is this ethical or not? You need to think about what we're doing with genes. We are manipulating the genes of the bacteria in order to save the lives of humans. And you need to decide for yourself whether you think this is an ethical process or not an ethical process, and where we have to draw the line. Those are questions that you need to resolve for yourself, and often you're asked to resolve them in an exam paper. So make sure that you think about that, you debate it with your friends, you debate it in class, so that you go into an exam being well prepared, not only on the science, but the ethics associated with the science as well. I hope you've enjoyed listening to our information today on gene therapy and genetic engineering. Go away, work very hard, and I'll see you next week.